Hello, welcome to Lecture 1 in the Principles of Cardiac Sonography 1 unit. This lecture is entitled Features of the Cardiovascular System and its Development. This lecture is also the first of the online lectures in the Graduate Diploma in Cardiac Sonography. I hope that you find this lecture and all the lectures in the course helpful. The contents of this lecture are 1. Anatomical terminology, 2. Features of the cardiovascular system, and 3. Cardiac development. Following the lecture, you'll be able to describe essential anatomical terminology, identify and link key features of the cardiovascular system, and discuss important aspects of normal cardiac development. The textbook to refer to, which is available via Views, is the Handbook of Cardiac Anatomy, Physiology and Devices, and the relevant chapters of the textbook are chapters 1, 2 and 3. Anatomical terminology. The anatomical position. In the standard anatomical position, a person is standing upright with the feet and palms facing forwards. Organs or structures within the body are described as if seen from this view. Anatomical planes. The body can be divided into 3 orthogonal, that is perpendicular, planes. The sagittal plane, a vertical plane which divides into left and right. The coronal plane, a vertical plane that divides into anterior and posterior. And the transverse plane, a horizontal plane which divides into superior and inferior. Various terms are used to describe the spatial relationship of anatomical structures. This slide provides a list of these terms which you're most likely familiar with. For example, the term superior is used to describe a structure nearer to the head than another. Anterior is used to describe a structure nearer to the front than another. Medial is a term used to describe a structure nearer to the median plane than another. And proximal, a structure closer to the point of origin than another. There are other terms on this table, and you'll need to be familiar with all of them. Echocardiographic terminology. Echocardiographic reference points. There are 2 primary cardiac reference points. 1. The apex, which is defined as the tip of the left ventricle. And 2. The base, which is a portion of the heart that forms its upper border, opposite the apex. Of note is the fact that all 4 heart valves lie in a similar transverse plane, which is nearer to the base of the heart than to the apex. Echocardiographic imaging planes differ slightly from the anatomical planes just outlined, although understanding anatomical planes will help you to discern key echocardiographic planes. The 4 orthogonal basic echocardiographic planes are the long axis, short axis, the 4-chamber and the 2-chamber planes. The long axis plane is similar to a sagittal plane within the left ventricle. The long axis plane within the left ventricle extends from the apex through the aortic valve. The short axis plane is similar to a transverse or horizontal plane. The short axis is perpendicular to the long axis plane and results in a cross-sectional view. The four-chamber plane, it's similar to a coronal plane. It divides the entire heart from base to apex to reveal all four chambers and is aligned with the mediolateral diameter of the mitral and tricuspid valves. And the two-chamber plane, which is similar to a sagittal plane through the left ventricle, and unlike the four-chamber view, it includes the left ventricle, left atrium and mitral valves only. Here are 4 echocardiographic images from the 4 basic imaging planes just described. The parasternal long axis image, the looping image, displays the heart from a plane that shows the long axis of the left ventricle and bisects the aortic and mitral valves, which help ensure image alignment. The looping short axis image shows a cross-sectional view of the left ventricle, the level of the papillary muscles. The looping four-chamber image displays an image from apex to base with all 4 chambers and is aligned with the mediolateral diameter of the mitral and tricuspid valves. The standard way to show apical images of the heart is with the ventricles at the top of the sector, that is, as though the probe were at the top of the screen. And the two-chamber image, looping at the bottom right, displays an image from apex to base with the left ventricle, mitral valve, and left atrium in view. The cardiovascular system. All the body's cells, which are organized into tissues and organs, need a constant supply of oxygen and nutrients, and also need to dispose of waste products. The primary function of the cardiovascular system is to provide a means of transport to help to ensure the normal functions of its cells, tissues, and organs. The 3 principal components of the cardiovascular system are blood, blood vessels, and the heart. Blood. Blood has 3 general functions. Firstly, transport. That is, via its components, blood enables the transport of elements around the body. Secondly, regulation. That is, blood helps to maintain a stable pH, temperature, and osmotic pressure. And thirdly, protection. Blood helps to defend from foreign materials and disease and excess blood loss. Blood is composed of cells suspended within plasma. Blood plasma, which is mostly water, makes up about 55% of blood by volume. The formed elements, the red blood cells, white blood cells, and platelets, which are all formed within the long bones of the body, are suspended within the plasma. As mentioned, plasma has a high water content and comprises about 55% of the blood by volume, and it suspends the other components of the blood. As well as the blood cells, these other components include plasma proteins, which enable the transport of lipids and hormones, vitamins and minerals. Plasma also absorbs and transports molecules and gases, and enables the transfer of and elimination of the thermal energy generated by the body's metabolic processes. Formed elements. Red blood cells, or erythrocytes, are produced in the bone marrow and are by far the most numerous cells within the blood. Red blood cells are shaped like biconcave discs and are about 8 micrometers in diameter. Hematocrit is a measure of the amount of red blood cells within the blood, and in men this is about 45%, and in women about 42%. The primary role of red blood cells is the transport of oxygen to make it available to cells around the body. Hemoglobin and iron-containing protein within red blood cells is the site at which oxygen binds. White blood cells. The primary role of white blood cells is the defense against foreign materials and abnormal cells. White blood cells are also formed in the bone marrow as well as in the lymph tissue. Unlike red blood cells, there are 5 different types of white blood cells, each with specific functions, which are beyond the scope of this lecture. The number of white blood cells varies. It varies depending on the changing needs of the body. Platelets. Platelets are irregularly shaped elements within the blood that have a lifespan of only 10 days or so. Normally, platelets circulate without adhering to blood vessel walls. However, when a blood vessel is damaged, the platelets aggregate and unite with collagen to adhere to the wall, to form a clot at the site of injury, and prevent blood loss. The second of the 3 principal components of the cardiovascular system are blood vessels. The primary function of blood vessels is to facilitate the passage of blood, and all vessel types have an important function. Arteries and arterioles carry blood from the heart. Capillaries are the site where the majority of exchange between the blood and tissues occurs. And veins and venules carry blood back to the heart. Normally, blood flow through the cardiovascular system is antegrade, that is, forward moving, because arterial pressure is greater than venous pressure. Arteries and arterioles are the more elastic type of blood vessels, as the blood within them is of a higher pressure. They have strong, thick, vascular walls with a relatively small vessel lumen. Veins and venules are the more distensible blood vessels, as the blood within them is of a lower pressure. They have thinner walls with a relatively large vessel lumen. Capillaries. Capillaries branch extensively throughout the body to bring blood within reach of every cell. Often no more than an endothelial cell layer thick, capillaries allow for the exchange of material via diffusion. Capillaries are the vessels where the majority of exchange between blood and tissues takes place. In 1628, British physiologist William Harvey accurately described the cardiovascular system as a closed-loop system, such that blood is pumped out of the heart through one set of vessels and returns to the heart via another. More specifically, the cardiovascular system is composed of 2 closed-loop systems, which both originate and return to the heart: the pulmonary and the systemic circulations. The pulmonary circulation is composed of the right heart and the blood vessels to and from the lungs. The systemic circulation includes the left heart and the blood vessels to and from the systemic organs. The two systems work in a series arrangement and normally circulate an identical volume of blood. The pulmonary circulation includes the right atrium, right ventricle, pulmonary arteries, lung capillaries and pulmonary veins. Normally, the pressure in the pulmonary circulation is significantly lower than the systemic circulation, with systolic and diastolic pressures of 25 and 10 millimeters of mercury, respectively. The systemic circulation includes the left atrium, left ventricle, systemic arteries, arterioles and capillaries, and systemic venules and veins. Normally, the pressure in the systemic circulation is significantly higher than in the pulmonary circulation. The heart. The heart is positioned in the center of the thorax and is the driving force of the circulation. The heart has two primary functions. One, to collect oxygen-poor blood and pump it to the lungs. And two, to collect oxygen-rich blood and pump it to the body. The cardiac cycle refers to the events that occur from one heartbeat to the next. In essence, there are two parts to each cardiac cycle. One, the contractile phase of the cardiac cycle, which is known as systole. And two, the relaxation phase of the cardiac cycle, which is known as diastole. The atria and ventricles both have diastolic and systolic phases. Atrial systole occurs before ventricular systole to ensure that blood moves in one direction. Most often when talking about cardiac systole, ventricular contraction, which is a more dynamic event, is the point of reference. So, unless otherwise stated, when talking I'll be referring to ventricular systole. When ventricular myocytes contract, pressure in the ventricles increases. When ventricular pressure exceeds pressure in the aorta or pulmonary artery, blood is forced out of the ventricle through the semilunar valves and into the great vessels. At the same time, the pressure in the ventricle exceeds pressure in the atria, and so the atrioventricular valves are forced shut to ensure that blood doesn't travel backwards. Ventricular systole is represented by the QRS complex on the ECG. When ventricular myocytes relax, pressure in the ventricles drops. When ventricular pressure falls below the pressure in the great arteries, blood is no longer forced out of the ventricle through the semilunar valves, and so these valves close. At the same time, as ventricular pressure falls to below pressure in the atria, the atrioventricular valves open. There are two parts to ventricular filling, which occurs in diastole. An early passive filling phase and a later active filling phase. The early phase is termed passive as the flow of blood is a result of changes in pressure, while the later phase is termed active as blood is forced forward as a result of atrial contraction. This diagram, which is taken from the textbook, provides some more information about the series of events that occur in one cardiac cycle, with particular reference to events on the left side of the heart. For now, I want to emphasize a select few events. 1. Electrical events precede mechanical events. For example, the QRS complex, which represents ventricular excitation, occurs prior to ventricular myocardial contraction. 2. Atrial excitation and contraction, indicated by the P wave, precede ventricular excitation and contraction, indicated by the QRS complex. 3. Changes in pressure dictate the opening and closing of the heart valves. For example, when left ventricular pressure rises to exceed aortic pressure, the aortic valve opens. And then, toward the end of systole, when left ventricular pressure falls below aortic pressure, the aortic valve closes. And 4. At rest, diastole is considerably longer than systole. With an increase in heart rate, the major change that occurs in the timing of the cardiac cycle is a reduction in the duration of diastole, whereas by necessity, the duration of systole remains largely unchanged. Normally, the heart functions very efficiently as a result of numerous well-functioning events, some of which I've described. The following 5 features are characteristic of normal cardiac function. 1. Synchrony. That is the regular and synchronous contraction of cardiac myocytes. 2. Normal full opening of the heart valves, which is opposed to valvular stenosis. 3. Normal full closure of the heart valves, which is opposed to valvular regurgitation. 4. Forceful ventricular contraction in systole. And 5. Adequate ventricular relaxation in diastole. This apical 4-chamber image of the heart features characteristics of normal heart function. The following can be observed. 1. The regular and synchronous contraction of the atria and ventricles. Note, as per convention, the atria are at the bottom of the screen, the ventricles at the top. Secondly, full opening of the heart valves, although only the mitral and tricuspid valves are seen in this image. Thirdly, closure of the mitral and tricuspid valves. Fourthly, forceful ventricular contraction in systole. And fifthly, adequate ventricular relaxation in diastole. The echocardiographic image on the left shows one example of how left ventricular systolic function can be measured. This measurement, which is a measure of what is termed the ejection fraction, has established normal and abnormal reference ranges, as indicated on the table on the right. What I aim to illustrate here is that echocardiography provides quantitative means of measuring ventricular contraction. In these echocardiographic images, LV diastolic function has been measured. Image A shows a measurement indicative of normal diastolic function. Images B, C, and D show varying degrees of diastolic dysfunction. What I aim to illustrate here is that echocardiography provides quantitative means of measuring ventricular relaxation. What is very important to understand is that both systole and diastole are essential components of the cardiac cycle. Regulation of cardiac function. Cardiac output, the product of heart rate and stroke volume, is a measure of the volume of blood in litres that the heart pumps per minute. If heart rate and stroke volume are known, cardiac output can be measured. For example, if the heart rate is 60 beats per minute and the stroke volume 100 mils per beat, cardiac output equals 60 times 100, which equals 6,000 millilitres per minute, that is 6 litres per minute. Normally, the cardiac output of an adult at rest is between 4 and 6 litres per minute. Cardiac output may increase 4 times or more with exercise. The cardiac output is regulated in two ways. One, via intrinsic control, and two, via autonomic control. Intrinsic regulation, that is, intrinsic control. Intrinsic regulation is related to changes in volume, as described by the Frank-Starling law of the heart. Simply stated, the Frank-Starling law indicates that the more the heart is stretched via increased return of blood, the greater the force of contraction. On the graph, it can be seen that with increased stretch, there is increased force up to a certain limit. Autonomic regulation. Autonomic regulation occurs as a result of control by the autonomic nervous system. The autonomic nervous system has two divisions, sympathetic and parasympathetic, which have antagonistic actions. Regulation via the sympathetic nervous system results in excitatory responses. Sympathetic regulation causes increases in heart rate and contractility. In opposition, regulation via the parasympathetic nervous system results in a calming effect. So, parasympathetic regulation causes a reduction in heart rate and contractility. There are important anatomic and chemical differences between the sympathetic and parasympathetic nervous systems, and many other important differences. While a detailed description of the autonomic nervous system is beyond the scope of this lecture, it's important to remember the following. 1. With regards to anatomy, in the sympathetic nervous system, postganglionic fibers arrive from the thoracic division of the sympathetic trunk, and from there innervate the heart. The parasympathetic nervous system's innervation is via neurons that branch from the vagus nerve. The vagus nerve, which travels down through the neck and into the thorax, is the 10th cranial nerve. Primary neurotransmitters. For the sympathetic nervous system, postganglionic fibers release noradrenaline, which activates adrenergic receptors to stimulate the heart. The parasympathetic nervous system uses acetylcholine as its primary neurotransmitter. Cardiac metabolism. The heart has its own metabolic needs. The heart, like the brain and skeletal muscles, needs a constant supply of blood. That is, the heart relies heavily on aerobic metabolism. At rest, it extracts 70 to 80% of the available oxygen from the coronary blood supply. Even minor ischemia, that is, even minor reduction in blood supply, can cause chest pain, that is, angina. Minor ischemia can result in changes on the ECG and a reduction in regional function. In contrast, other organs like the lungs, kidneys and skin can go for longer periods without blood supply. Cardiac development. The human heart is the first organ to form and function. There are 4 major embryonic regions for cardiac development: the primary heart field, the secondary heart field, the cardiac neural crest, and proepicardial organ. Cardiac development is complex, and each region has a unique contribution. The primary and secondary heart fields. The primary heart field is the result of the fusion of 3 primary germ layers. A linear heart tube develops in the primary heart field. By day 28 of human development, the linear heart tube is developed, and by day 22, the linear heart tube begins to beat. The linear heart tube is composed of 4 layers: endocardium, cardiac jelly, myocardium, and epicardium. Tissue is added to the linear heart tube from the secondary heart field from day 22. Heart looping and continued growth occur during days 22 to 28 of development. And by the 28th day of development, the chambers of the heart are in position. The cardiac neural crest and proepicardial organ. Septation of the heart occurs after looping and when the chambers are in the correct position at about 28 days of development. Ventricular septation is complete after 9 weeks of development. Functional septation of the atria, however, does not occur until after birth. Cardiac neural crest cells are essential for septation of the heart's outflow tract and ventricles. The proepicardial organ is key in the development of the coronary vasculature. There are important differences between the fetal and infant circulation. Prior to birth, blood can cross from the right to the left via 1. The patent foramen ovale at the level of the atria, and 2. Via the ductus arteriosus, which links the pulmonary artery and the descending aorta. Before birth, the baby depends on its mother for the supply of oxygen-rich blood via the placenta, as the lungs are not yet functional. After birth, the umbilical blood supply is interrupted. Plus, with the first breath, the resistance in the lungs falls, allowing blood to flow into the lungs and to drain into the left atrium, which means a reversal in pressure occurs, forcing the foramen ovale to close and therefore helping to complete atrial septation. This process, together with the closure of the ductus arteriosus, separates the right and left sides of the heart. Cardiac maturation. By the 11th week of development, the heart is fully formed and functional. the fetal heart continues to grow, primarily by cell division. After birth, the heart continues to grow. However, after birth, in contrast to maturation of the fetal heart, growth is primarily by cell hypertrophy. This table shows 5 developmental milestones and their time points. On day 22, the linear heart tube begins to beat. From days 22 to 28, heart looping occurs. At 9 weeks, ventricular septation is complete. By 11 weeks, the heart is fully formed and functional. However, only after birth does functional atrial septation occur. In conclusion, the first part of this lecture included important aspects of anatomical terminology, such as the 3 orthogonal planes used to divide the body, as well as the 4 essential echocardiographic imaging planes. Next, we considered the cardiovascular system. Its essential aspects and components were described. These include blood and its primary functions and constituents, blood vessels, their organization and primary function, and some aspects of the heart and its regulation were also described. And finally, we considered cardiac development, the fact that the heart is the first organ to form and function. The 4 major regions for cardiac development and the unique contribution of each region was described, as well as the difference in growth patterns between the fetal heart and the heart following birth.